Okay. Good evening, everyone. My name is Mia, and I and Nuria are students of Istanbul Technical University, majoring in molecular biology and genetics. Simultaneously, we're part of Beeware, a group of four undergraduate students that aim on raising awareness on cystic fibrosis.
And I and
In this
Nuria are
group,
students of Istanbul Technical University, majoring in molecular biology and genetics.
I think there is a problem. There should, but it's okay. We can continue. Life things happen. In the scope of our project, we aim on raising awareness of cystic fibrosis, as I said before. So we're doing our third interview. We have the honor of having Katja Koeppen, a research scientist from Dartmouth University. She kindly accepted to be with us today and show us a bit of her work on the lab. Said so, today's talk, it will be focused on host pathogen communication through extracellular vesicles, and it will all be in the context of lung infections that affect cystic fibrosis patients. For the Turkish version of this introduction, you can listen to Rüya.
Herkese iyi akşamlar. Ben Beeware takımından Rüya Aslan. Mira Tafa'da Beeware takımından. İstanbul Teknik Üniversitesi'nde moleküler biyoloji ve genetik okuyan ve kistik fibrozis hastalığı konusunda farkındalık yaratmayı amaçlayan 4 kişilik bir takımız. 3. röportajımızda Dartmouth Üniversitesi'nde araştırma görevlisi olan Katja Koeppen ile tanışacağız. Katja Koeppen'e bugün zamanını bize ayırdığı için çok teşekkür ederiz. Konuşmanın konusu kistik fibrozis hastalarının akciğer enfeksiyonu bağlamında hücre dışı veziküler yoluyla konak patojen iletişimiyle ilgilidir. Hepinize iyi seyirler.
Without further ado, we're giving our word to our special guest of the day.
All right. Thank you so much for the kind introduction, and thank you all for your interest in my work. I'm very excited to talk today about the projects that I've been working on in the recent years, and I do want to start out with a brief introduction that cystic fibrosis is a multi-organ disease, and it is caused by mutations in the chloride channel CFTR, cystic fibrosis transmembrane conductance regulator. And originally, 50 or so years ago, cystic fibrosis was first and foremost a disease of the gastrointestinal tract, and infants born with cystic fibrosis would often not live past the first year of life due to the inability to absorb nutrients properly. And with the invention of giving pancreatic enzymes to CF patients, the gastrointestinal problems were alleviated. And nowadays, one of the main problems for cystic fibrosis patients is lung infection and ultimately lung failure. However, as treatments for CF have become more advanced, we have realized that cystic fibrosis affects additional organs besides the gastrointestinal system or the lungs. For example, it affects the bones and the liver, and can also affect the male reproductive system. Now, my research primarily focuses on the lungs and the airways. And this on the right is a cross-section of a normal airway. And when CFTR is functional, it pumps chloride ions out of the cell, and that allows water to follow, and it hydrates the airways. So the result in the healthy state is a hydrated airway, a hydrated mucus layer. And this mucus layer serves to constantly trap bacteria and other pathogens that are inhaled and transport them out of the lung. And this is the very process that is impaired in CF patients. So on the very far right here, where a mutant CFTR channel fails to pump adequate amounts of chloride out of the cell, it leads to the buildup of dehydrated and sticky mucus. And this mucus cannot efficiently be transported out of the lung anymore, and it serves as a breeding ground for trapped bacteria and other pathogens such as virus or fungi. And that really is one of the hallmarks of CF, this inability to clear pathogens from the airways. Now, let's take a closer look at the kinds of pathogens that are commonly found in the lungs of cystic fibrosis patients. This graph is data from the US CF patient registry, and it is showing the prevalence of various respiratory pathogens stratified by age. So on the far left are very young patients of less than 2 years of age, and on the far right are older CF patients of 45 or above. And the two most common bacteria that are detected in the lungs of CF patients are Staphylococcus aureus in green and Pseudomonas aeruginosa in light blue. And if you look on the left in the young patients, almost 60% of young patients have Staphylococcus aureus in their lungs. And Pseudomonas aeruginosa is less often detected in very young children. But as patients age, Pseudomonas becomes more and more prevalent. And ultimately, over 70% of adults with cystic fibrosis are infected with Pseudomonas aeruginosa. And one thing that is important to note is that these bacterial lung infections are often chronic. So unlike lung infection in a non-CF person with a healthy immune system where they might get temporary pneumonia, these lung infections in CF patients are often chronic and persist over many years. And even antibiotic treatment usually does not eradicate the bacteria. Although the patients often do feel better after antibiotic treatment, we now know that it is not due to the bacteria being eradicated, but rather we think it is that the antibiotics make the bacteria less virulent and that improves the health of the patient, but it is not due to actually defeating the bacteria with the antibiotics, because they have a way of forming communities called biofilms, where many of them aggregate together in a matrix, and they become very resistant to antibiotic treatment. So that is one of the huge problems and challenges nowadays, how to actually treat these highly antibiotic-resistant chronic bacterial infections. And that's where my research comes in. Now, one more thing I want to give as a background before diving into my particular research is related to the innate immune response that normally happens in response to bacterial infection. So this would be what typically happens in a non-CF person if a bacterial infection is detected. So here we have airway epithelial cells, and in red is Pseudomonas aeruginosa. And the airway epithelial cells have a receptor called TLR4, and this is just a binding pocket that can recognize bacteria, such as Pseudomonas aeruginosa, and bind them. And in response to this binding of bacteria, a signaling cascade is triggered that leads to the release of cytokines from the epithelial cells into the bloodstream. And these cytokines actually activate macrophages and neutrophils and call them to the site of infection. So this is a very powerful first signal from the airway cell to professional immune cells such as macrophages and neutrophils. And now what do the professional immune cells do? So on the right in the center is a macrophage. And the macrophages, just like the epithelial cells, also have the TLR4 receptor. They can also recognize and bind bacteria, and they can amplify the signal and also secrete these chemical molecules called cytokines that will in turn activate and recruit even more macrophages and neutrophils. And this serves basically as an amplification signal. And one very, very important function of the macrophages is that they can actually phagocytose, which means take up bacteria and kill them. So they are real bacteria eating and killing machines. And this is really how a healthy immune system deals with a bacterial infection, that the first line of defense is to have macrophages and also neutrophils. They can also take up bacteria, eat the bacteria, and kill them. And that is very, very important. And that is one of the aspects that is also impaired in cystic fibrosis patients. So here on this next slide, I'm going to address some of the immune defects in people with cystic fibrosis. And one is on the upper left, that the sensing of bacteria through the TLR4 receptor is actually reduced in CF airway epithelial cells. So this detection doesn't work properly. And so initially they can't mount an appropriate response, and the response of the CF airway cells is attenuated as opposed to the wild type. What also is the case in CF patients is that they have an impaired activation of their macrophages. So the whole process of detecting bacteria and then activating macrophages to kill the bacteria is impaired and attenuated. It just doesn't work as efficiently in CF patients. And this is all due to the malfunction of CFTR, the chloride channel. And now it is now also becoming apparent that the cytokine secretion by the CF macrophages is also altered in comparison to wild-type macrophages, where some cytokines are actually secreted more than you would see in wild-type and others less. And finally, this very, very important step of phagocytosis, taking up bacteria and killing them, is also impaired and reduced in the CF macrophages. So you can see that many different components come together that collectively explain why CF patients have a hard time eradicating bacteria from their airways. And now, in order to better understand where my research comes in, I want to give some final background as to what extracellular vesicles actually are. You've seen in my title, it is about communication between the host and pathogens through extracellular vesicles. But what are these vesicles? So for scale, if the top line is an airway epithelial cells, that's on average about 20 micrometers. In comparison, a Pseudomonas aeruginosa cell is about a tenth of that, around 2 micrometers. And the EVs that are produced by Pseudomonas are also referred to as OMVs, or outer membrane vesicles, because they are budding off the outer membrane of the bacteria. And they range in size between 50 and 250 nanometers. So this little dot on the left is supposed to give you a sense of the scale. And then the human airway cells can also produce vesicles, and they can produce two different types of vesicles, well, at least two different types of vesicles that are commonly detected. And one are exosomes that are 50 to 150 nanometers in size. And then there's also microvesicles, which have a large range of sizes, but about 100 to 1,000 nanometers. And just so that you get a better sense of what this all looks like, this image is a Pseudomonas aeruginosa cell, and all the little round blebs are outer membrane vesicles that are budding off the membrane. And on the right here is a cartoon diagram of a eukaryotic cell. So this could be a human cell, could be an airway cell. And you see the two types of vesicles. The microvesicles are just like the outer membrane vesicles in that they bud off the outer membrane of the cell. And the exosomes are produced a little differently. They are made in this so-called endosomal pathway and are then released en masse, as you can see in this diagram. But ultimately, we're not so concerned about the origin of these vesicles as we are interested in their function in communicating. And it has been recently found that there's a lot of selective packaging of proteins, but also RNA into extracellular vesicles. And the figure on the left, on the lower left, shows a example of a human EV, an exosomal EV, and it has all sorts of membrane proteins, but also intracellular proteins contained inside of it. A big component is also microRNAs, mRNAs. It can also contain DNA. So all this cargo has been detected in the vesicles. And what has also been shown is that these extracellular vesicles can transfer their cargo to a recipient cell. And this has been shown for the extracellular vesicles of human cells, that one cell type can produce them and they can then be delivered to a different type of cell within the human body. And it has also been shown that the bacterial outer membrane vesicles get delivered to airway epithelial cells. So this is like a trans kingdom communication, if you so will, where the bacterial vesicles deliver their cargo to the human host cells and affect the cells in that way. And now this is where my research really comes in. So the big picture is that we are now finding, and this is all relatively recent research in the last 5 years or so, we are finding that these extracellular vesicles play a very important role in communication between host cells and pathogens. And this communication is bidirectional. And that's all pretty much pretty new cutting edge research. So in this publication 5 years ago, we showed that Pseudomonas aeruginosa produces outer membrane vesicles that contain a short RNA, and that this short RNA, when it gets delivered to airway cells, it interferes with the immune response. So IL-8 is one of the cytokines that I showed you about previously that usually get released when bacteria are detected. And the cell on the right here is supposed to be a neutrophil. So what usually happens when bacteria are detected is that the airway cells secrete cytokines such as IL-8, and that recruits neutrophils. But we showed that the bacterial small RNA that gets delivered in these outer membrane vesicles, that reduces IL-8 secretion and it reduces neutrophil recruitment. So it basically interferes with the host immune response. Another finding from a very recent paper earlier this year was that exposing airway epithelial cells to Pseudomonas and then collecting the extracellular vesicles that are secreted by the airway cells in response to having seen the bacteria. If you take these EVs and expose macrophages to these EVs, it actually leads to increased cytokine secretion by macrophages. So this would be an example of EVs actually helping mediate an innate immune response. And I will talk about this a little more in a minute. But finally, I do want to share the third part of the story, which I'm actually really excited about. And this is, I think this paper is supposed to come out sometime this week. So this was showing that microRNAs that are contained in vesicles secreted by airway epithelial cells if Pseudomonas aeruginosa is exposed to these human EVs, the microRNA actually gets delivered and it reduces their ability to form these antibiotic-resistant biofilms, and it also lowers their antibiotic resistance. So we think that this, and this is a new mechanism because it hasn't been shown before that human microRNAs in EVs can affect bacteria. And we are pretty excited about this and thinking that there might be some promise of therapeutic potential in the future. But for the remainder of this presentation, I will just focus on the second story here, because there is a very interesting difference between CF and wild-type macrophages and in their response to extracellular vesicles. And what we found is that the response of CF macrophages to EVs is attenuated. So just like I mentioned previously, that CF macrophages have attenuated ability, for instance, to phagocytose and kill bacteria, they also seem to be impaired in their response to extracellular vesicles. And this diagram here shows at the bottom are airway epithelial cells, either wild type or CF, and they release extracellular vesicles, and we have then exposed either wild type or CF primary monocyte-derived macrophages to these EVs. And what we noticed is that you do get some cytokine secretion by the wild type macrophages, and you get many changes in their gene expression at the mRNA level. But looking at the CF macrophages, there was no significant increase in cytokine secretion and an attenuated gene expression response. So whatever the wild type does in response to EVs, the CF macrophages do less of it. And this was even more pronounced in the case of EVs secreted by airway cells after they've been exposed to Pseudomonas. So PA14 is a strain of Pseudomonas in the lower right here. And we've exposed either the wild type or the CF airway cells to Pseudomonas, and we have then collected extracellular vesicles secreted by the airway cells. And one first difference we noticed was that the wild type airway cells secrete a lot more, about twice as many vesicles than the CF airway cells. So this is another defect in CF, that it seems that their ability to secrete these extracellular vesicles in response to sensing a bacterium, that this response is impaired in CF airway cells. But then if we take the same number of extracellular vesicles and expose wild type or CF macrophages, again, we get a significantly attenuated response from the CF macrophages compared to the wild type. The wild type responds with a very massive induction of secretion of different pro-inflammatory cytokines, but also tissue repair factors. And this is all attenuated in the CF macrophages. So we believe that this also helps contribute to chronic bacterial infections, that this attenuated response actually leads to a failure to properly eradicate the bacteria. And well, with that, I would like to thank all my colleagues in the Stanton Lab and my collaborators at Dartmouth. And our funding is from NIH and from the Cystic Fibrosis Foundation. And I think with that, I'm happy to answer any questions that you may have.
Well, thank you so much for your presentation. We're going to wait a bit so that perhaps someone can write to the chat box. But on the meantime, I had something like, what would happen, or more exactly, what happened to those CF patients in the time when we had no antibiotics? Like, I'm believing that their life expectancy was extremely low, right? It should have, because...
Yes, it was originally extremely low, as I mentioned, due to the inability to absorb nutrients. But after that problem was solved by giving patients pancreatic enzymes to help with their digestion, oftentimes CF patients would still not reach adulthood because of lung infection. So having antibiotics has definitely helped some, but it hasn't completely solved the problem. I would say in recent years, the new Vertex drugs that are given to not all patients, because it depends on the mutation CFTR whether a patient is eligible, but those who are, it seems that they have greatly benefited from the latest generation of drugs. But even so, what we're finding in older patients who have chronic bacterial infection, the drugs don't always clear up those infections. So it is basically, I think, a constant tug of war between the host and the bacterium. And yeah, but definitely having more treatment options, the life expectancy of CF patients is way up. It's now, I think, around 40 years and probably even going to be higher with the new drugs.
We have a question from Begüm Aksoy. She says, does the attenuated response of neutrophils have also an identified genetic background? Like, is this response kind of related to genetics?
Yes, we do think that it is related to the defect in CFTR. And we don't fully understand all of it, though in the case of the attenuated phagocytosis, the attenuated ability to take up and kill the bacteria, that is directly related to CFTR, because CFTR is basically required to regulate this process of uptake and of killing.
Apparently there's no other question. I thought there would be more, but...
Well, you can always feel free to email me later if additional questions come up.
Thank you. I really appreciate it. Indeed, it's been a really nice communication. Thank you.
Yes, you're very welcome, and I wish you the best with your project.
Thank you so much. Like, since there's apparently no other comment or question, I guess that would be for today. Oh, there's another one. So,
Sure.
how the content of EVs, extracellular vesicles, change when the pathogen changes? That would be the question.
The question is whether exposing airway cells to different pathogens would change the contents of the EVs, or actually just exposing the airway cells. Well, what I can say is exposing the airway cells to Pseudomonas definitely changes the contents of the EVs that are secreted by the airway cells. And I haven't looked at any other pathogens, but I would basically suspect that you would get changes in response to other pathogens as well in the contents of EVs. And there might be differences in how different pathogens affect the EV content.
I guess Begüm got her response. So because she cannot join the video conference, she can only chat to us. So I'm assuming that she already got her response. And I believe this is it for today. Thanks a lot for dedicating your time to us. We're really honored. It was a really qualitative presentation. Me, as a molecular immunology student, I learned a lot, and I really enjoyed it, as I have taken the class. So
wonderful. Well, thank you so much, and I was glad to help. And feel free to email me if questions come up later.
Thank you. So that would be it, and thank you all of you for listening to us today, for being live on YouTube, even though we cannot communicate directly. Thank you for being there as viewers.
Yayınımıza katılan herkese çok teşekkürler. Sonraki yayınlarda görüşmek üzere.
Thanks a lot. Goodbye.
Bye.